Selection of host organism and vector system. CIP, or calf intestinal phosphatase, removes phosphate groups from DNA ends by hydrolyzing phosphate monomers, such as the 5' hydroxyl termini, to prevent vector self-ligation during cloning. This dephosphorylation eliminates the 5' phosphate needed for DNA ligase to join vector ends, ensuring only recombinant DNA forms. Vectors are self-replicating DNA tools that deliver DNA fragments into cells, including plasmids, bacteriophages, eukaryotic viruses, and artificial chromosomes. They need a small size, between 2 and 5 kilobase pairs, for easy handling and replication, unique restriction sites for precise DNA insertion, at least one selectable marker gene like antibiotic resistance to identify transformed cells, and replicons that match the host. Although binary shuttle vectors can work in multiple hosts with different replicons, vectors serve different purposes. Cloning vectors store and amplify DNA. Expression vectors produce recombinant proteins, and shuttle vectors replicate across organisms. Cloning vectors store and amplify DNA for later use, like sequencing, using a host-specific replicon such as the PBR322, origin for E. coli. A cloning site, often called a multiple cloning site, or MCAs, with unique restriction sites like Acora or BAMI, and a selectable marker. For example, ampicillin resistance from the BLA gene. Examples of cloning vectors include plasmids, which hold 1 to 10 kilobase pair inserts and are small, high-copy tools like PUC. Bacteriophages or viruses like lambda-phage that carry 10 to 25 kilobase pair inserts. Phagemids that are plasmid phage hybrids for single-stranded DNA useful in sequencing. Cosmids that are plasmid phage hybrids with CoA sites holding up to 45 kilobase pairs, BASE or bacterial artificial chromosomes that handle up to 300 kilobase pairs with low copy number and stability for large DNA, and YAC or yeast artificial chromosomes that manage 0.5 to 1.5 megabase pairs by mimicking eukaryotic chromosomes. Expression vectors produce recombinant proteins from inserted DNA and contain regulatory elements like a promoter before the cloning site to start transcription and control protein levels, with promoter regulation often based on the E. coli lac operon, where the loci repressor blocks expression until induced by L-lactose naturally or by IPDG. Isopropyl beta-D-thiogalactoside in labs for precise control. These vectors can be plasmid-based for bacteria or virus-based for eukaryotes and work in either prokaryotes or eukaryotes depending on their design. Shuttle vectors replicate in multiple organisms for flexible cloning workflows, allowing DNA to be cloned in E. coli and then transferred to a eukaryotic host like yeast or mammalian cells. They feature bacterial elements, such as an E. coli origin, like PUC ori, and a resistance gene, like kanamycin, alongside eukaryotic elements including a promoter, a poly-A signal for mRNA stability, and a Kozak sequence, GCCAG, for translation start, with replication depending on origins like PUC ori for high copy, and E. coli or tuma ori for yeast. Plasmids are extrachromosomal, independently replicating circular DNA segments that are non-essential and commonly used in prokaryotes and yeast as plasmid vectors. Their modular structure includes a replicon, which consists of a replication origin and nearby regulatory sequences specific to bacteria, yeast, or eukaryotes, initiating replication while controlling copy number. They also feature an MCS, or multiple cloning site, with restriction enzyme cleavage sites for DNA insertion, alongside selectable markers to identify transformed cells. These markers include antibiotic resistance, which differentiates cells with vector or recombinant DNA from those without, using drugs like ampicillin, tetracycline, kanamycin, chloramphenicol, or neomycin. Ampicillin resistance, for instance, comes from a penicillin derivative that inhibits bacterial cell wall biosynthesis by blocking transpeptidase, an enzyme critical for peptidoglycan or murin formation. Another marker is auxotrophic selection, which relies on enzymes essential for metabolism and uses dropout media lacking specific nutrients like adenine, histidine, leucine, lysine, tryptophan, or uracil. Though this method is expensive, time-consuming, and limited to specific cell lines since auxotrophs cannot synthesize certain organic compounds needed for growth. Blue-white selection distinguishes recombinant from empty vectors through beta-galactosidase, alpha complementation, where the LACH gene encodes beta-galactosidase to cleave X-gal and produce a blue color, but the MCA is within the LACH alpha subdomain. So DNA insertion disrupts LAC-A, preventing enzyme activity. IPTGG induces lac operon expression, leading to white colonies with recombinant DNA, where LAC is disrupted and no blue color forms, versus blue colonies with empty vectors, where intact LACH allows beta-galactosidase to cleave X-gal. The genetic code operates in triplets, and shifts in the reading frame can occur, while growth from one colony yields about 10 million cells overnight. Though blue-white selection is rarely used in labs today due to its labor intensity, with PCR and sequencing offering simpler alternatives. Main plasmid types, which overlap in categories, include F plasmids, which are conjugative and enable bacterial conjugation for sexual reproduction. R plasmids, which carry resistance genes transferable via horizontal gene transfer, and col plasmids, specific to E. coli, containing colicin genes that produce bacteriocins or toxins targeting other bacterial strains. Plasmid replication is mostly unidirectional, starting from one replication fork at the origin, with copy number influenced by the replication origin type and plasmid size, including any insert. High copy number aids cloning, but may complicate expression depending on the protein produced. Specific plasmid vectors, like PBR322, are E. coli-compatible cloning vectors with a low copy number, featuring 40 unique restriction sites and resistance genes for ampicillin, with 6 sites, and tetracycline, with 11 sites, where insert integration into these resistance gene regions allows selection by loss of resistance function. The PUC vector family contains the piombone replication origin, an ampicillin resistance gene, and part of the LACH gene, called the LACH alpha fragment, with an MCS offering dual selection through primary antibiotic resistance and secondary blue-white screening via LACH alpha. Plasmids with identical replication origins are incompatible, meaning only one persists per cell unless selectable markers differ, as competition for replication factors causes one to dominate. Expression plasmids are practical for cloning directly in expression or shuttle vectors and require compatible bacterial strains, such as those expressing 7 polymerase, with elements like the 7 promoter regulating transcription, active only with 7 RNA polymerase in strains like BL21 to 3 E. coli endo, induced by IPTG to drive high mRNA production, and RBS, or ribosome binding site, with the Shine-Dalgarno sequence, enabling ribosome binding for translation, and a terminator sequence, stopping transcription to release nRNA. Source DNA isolation and preparation begins with cDNA isolation, where complementary DNA or cDNA is synthesized from RNA and lacks intron. The process starts with an oligo, DT, primer binding to the 3' polyetail of nRNA, followed by reverse transcriptase synthesizing the first DNA strand from the nRNA template, after which RNase H degrades the RNA, and DNA polymerase I synthesizes the second DNA strand, with PCR then amplifying the cDNA using complementary oligonucleotide primers that include restriction sites, before the digested plasmid is ligated and transformed into bacteria. DNA amplification and modification happen through PCR, or polymerase chain reaction, conducted in a programmable thermostat with a 20 to 50 microliter volume, using thermostable DNA polymerases to amplify DNA, allowing modifications via oligoprimers such as restriction sites, start and stop codon, affinity tags like polyhis, and point mutations including deletions, insertions, or substitution, with oligo design being critical for specific amplification and modification. DNA isolation from agarose gel involves electrophoresis on a horizontal gel made of agarose from red algae. Visualization with a fluorescent intercalating dye that's mutagenic to see the DNA, and a sample rerun for verification and molar ratio determination. Insert preparation starts with selecting the DNA segment to study, requiring a genomic or cDNA library for an unknown sequence, or obtainable from DNA databases for a known sequence, with 3 methods available: self-production from genomic DNA or RNA samples, synthesis ordered synthetically, which takes minimal effort and time, but is expensive, or using a clone database like AdGene to obtain recombinant DNA containing the insert, then excising it with restriction enzymes or subcloning via PCR. Depending on whether you're cloning a gene fragment, or cDNA, which is a reverse transcribed NNA copy for cDNA libraries or expression construct. For a genomic DNA insert, if working with a gene, you need a species-specific genomic library or isolated genomic DNA. Amplifying specific DNA from a gene library or isolated genomic DNA using PCR with primers that can include 5' restriction endonuclease site. Running the PCR product on agarose gel for size verification. Isolating the DNA. Digesting the isolated insert with a restriction endonuclease to create sticky ends for ligation with a complementary DNA fragment, and purifying the DNA using a silica column to remove enzymes and nucleotide debris, preparing it for ligation. A cDNA insert, used to study the protein encoded by the gene, requires cDNA synthesis or synthetic DNA, starting with selecting tissue or cell culture with high miRNA expression of the target gene, then isolating total RNA. For cDNA library, miRNA is isolated from total RNA using the POLIA tail for one-step affinity chromatography with 1 oligodeity cellulose binding nRNA, followed by cDNA synthesis with reverse transcriptase using oligoDT primers. While for expression constructs, cDNA is synthesized directly from total RNA and amplified via PCR, with primers containing 5' restriction endonuclease sites. Gene libraries include genomic DNA, or gDNA, libraries, with 1 per species, using vectors like lambda-phage, cosmids, BAC, or YACE, and cDNA libraries synthesized from RNA to DNA, using reverse transcriptase, with multiple cDNA libraries per species that are tissue-specific. Where sequences can be found in databases, like PubMed, UniProt, ExPC, or JustBio, and plasmid databases, like AdGene, provide sequenced, published plasmid construct. The plasmid preparation workflow involves ordering a plasmid construct from a database, isolating plasmid DNA from a bacterial strain, and verifying it by sequencing, amplifying the insert via PCR with appropriate restriction sites, digesting the target plasmid and PCR product, ligating the insert into the target plasmid, transforming it into bacteria, and isolating the DNA for final sequencing verification. Cloning involves restriction endonucleases, which originate from the bacterial restriction modification system, a bacterial immune system, alongside CRISPR-Cas, protecting against foreign DNA by hydrolyzing its specific recognition sites of 4 to 8 nucleotides. While DNA methylases mark the bacterium's own DNA at these sites to prevent cleavage, with specificity, typically for palindromic sequences of 4 to 8 bases that read the same. Forward and backward, cutting both DNA strands at or near the recognition site to produce sticky ends from offset cuts, creating overhanging ends that pair via Watson-Crick. Base pairing or blunt ends from cuts at opposite phosphodiester bonds, producing flat ends, allowing DNA from different sources cleaved with the same enzyme to generate compatible fragments that DNA ligase can covalently join, as seen with E. coli from Escherichia coli, strain Rhy13. Enzyme first, recognizing GAT to produce 5' overhanging sticky ends, one of over 3,000 identified restriction enzymes with over 200 unique recognition sites. Structured as dimeric proteins requiring magnesium ions to hydrolyze phosphodiester bonds in double-stranded DNA. DNA ligase facilitates ligation by forming phosphodiester bonds to covalently link 2 DNA molecules or ends, integrating the target DNA sequence into a vector. Using ATP as a cofactor in eukaryotes, viruses, and bacteriophages, or NAD+ in bacteria, with T4 DNA ligase from T4 bacteriophage, infected E. coli, being common. Relying on ATP and capable of ligating blunt ends, succeeding when DNA ends are close, as sticky ends form weak hydrogen bonds, aiding ligase binding, while blunt ends lack this interaction and reduce efficiency, requiring known concentrations of insert and vector DNA, often with CYP or calf intestinal alkaline phosphatase, reducing empty vector self-ligation by removing 5' phosphate groups from linearized vectors after single enzyme digestion to prevent vector self-closure or concatenation formation. Tau cloning uses linearized Tau cloning vectors with 3' overhanging thymine sticky ends pairing with 3' overhanging adenine ends on the insert, prepared by amplifying with a DNA polymerase like Taq, which lacks 5' to 3' proofreading exonuclease activity, but has template-independent polymerase activity as a terminal transferase, adding an adenine nucleotide to the 3' ends, followed by post-transformation screening via blue-white selection. LIC, or ligation-independent cloning, avoids DNA ligase and restriction enzymes, rely on the 3' to 5' exonuclease activity of DNA polymerases, like T4 or PFU. Create long sticky ends on both insert and linearized vector, amplifying the insert via PCR with primers designed for overhangs of 15 nucleotides, or more compatible with Ward vector ends, where overhangs longer than 15 nucleotides hold the construct together in the cell without ligation, transforming into competent cells, where endogenous DNA ligase forms phosphodiester bond. Tau-topo cloning, similar to cloning, uses a linearized vector with covalently attached vaccinia virus DNA topoisomerase I at the 3' end to facilitate ligation, amplifying the insert with Taq polymerase to add 3' adenine pairing with the vector's 3' thymine overhang, requiring a specialized Taq cloning vector with topoisomerase I acting as both restriction enzyme and ligase, needing no separate ligase or restriction enzyme. Zero blunt topo cloning uses a linearized vector with covalently bound topoisomerase I to ligate blunt-ended PCR products from high-fidelity polymerases like FU or fusion, rather than Taq, featuring non-directional ligation without ligase, while directional topo cloning uses a topoisomerase I-bound vector to ligate blunt PCR products with a 5' CAC sequence, ensuring directional insertion at the CAF-GTDZ site, also without ligase. Gateway cloning enables ligation without restriction digestion via site-specific recombination between lambda-phage ATP and bacterial ATP sites, with a BP reaction introducing ATP and ATP sites into the insert via primers and lambda-phage integrase opening ATP to join ATP, forming ATL and AYER hybrid sites, and the reverse reaction using integrase to recognize ATL and ATR, excising and recircularizing the phage DNA to restore ATP and ATL. Ideal for transferring an insert into multiple vectors, such as from an entry vector with ATLO and ALTO2 to destination vectors with ATRO2 via LR cloning, offering high efficiency at 99% or more in 1 hour at room temperature, preserving the reading frame, and proving useful for transgenics and complex subcloning like building large inserts from small fragments, incorporating negative resistance genes, and targeting vectors without restriction digestion. Transformation into an intermediate cell line for clone isolation uses bacterial transformation with competent cells capable of uptake DNA from the medium, employing heat shock and cooling for E. coli, which has 2 membranes, followed by DNA isolation via Miniprep for small-scale plasmid DNA isolation, or Maxiprep for large-scale plasmid DNA isolation. Transformation into the target cell line with shuttle vectors begins with transformation into bacterial target cells, where bacteria uptake DNA molecules like plasmid DNA from their environment, a common method to introduce recombinant DNA into bacterial cells. Starting with competent cells prepared by incubation in calcium chloride solution, which facilitates plasmid DNA binding to the bacterial cell wall's lipopolysaccharide, as positively charged calcium ions attract negatively charged DNA, followed by heat shock increasing membrane fluidity to allow DNA entry into the cytoplasm. Regeneration in nutrient-rich medium for cell recovery, and selection on selective media to identify transformants. Electroporation, used for yeast, bacteria, and mammalian cells, is the most efficient method for introducing CNA, applicable to nearly all cell types with high efficiency. Though excessive voltage or duration can kill cells, and efficiency depends on local transmembrane voltage, with an electroporator generating an electromagnetic field in a cell. Suspension mixed with DNA, placed in a cuvette with electrodes on both sides, applying high voltage of 10,000 to 100,000 volts per centimeter, from microseconds to milliseconds, temporarily, creating pores in the phospholipid bilayer and cell wall, allowing DNA entry due to high extracellular plasmid concentration, with pores closing when the membrane charge dissipates and the lipid bilayer reseals, followed by immediate placement in medium for regeneration, where surviving cells often contain the plasmid at high rate. Transfection, similar to transformation, but targeting mammalian eukaryotic cells, using eukaryotic expression vectors, like plasmids to express proteins temporarily or stably. With selectable markers, faces challenges with efficiency depending on DNA composition, solution pH, and cell membrane condition. Using chemical methods that bind negatively charged DNA to cationic carriers for cellular uptake, such as cationic lipids like lipofectamine forming liposomes encapsulating DNA that fuse with the cell membrane to release DNA into the cytoplasm. Calcium phosphate, a cost-effective method where calcium chloride is added to a phosphate buffered solution like heaps with DNA to form a precipitate binding DNA on its surface for uptake via endocytosis, and cationic polymers like D-dextrose or polyethylamine binding negatively charged DNA for entry via endocytosis, with transfection methods combining chemical and physical approaches, including turbofect reagent, a synthetic cationic polymer forming a stable, positively charged complex with plasmid DNA to prevent degradation, adhering to the cell membrane, and entering via endocytosis, possibly macropinocytosis, with protein expression occurring 12 to 72 hours post-entry, though the mechanism remains unclear. And lipofectamine 2000, a cationic liposome, forming complexes with negatively charged DNA or RNA, fusing with the negatively charged cell membrane to deliver nucleic acids into the cytoplasm, optimized by factors like cell type varying by target, plasmid and reagent depending on specific construct, DNA quality requiring high purity with an odd 260-280 ratio greater than 1.8 and endotoxin-free, cell density at 70 to 90% confluence at transfection. Reagent interactions with antibiotics or serum components affecting efficiency and viability, DNA to reagent ratio impacting efficiency and survival. An expression vector, influencing protein expression levels through promoter strength, rather than the transfection method. Assessing transfection efficiency with FACS, or flow cytometry, analyzes particles like cells, beads, or bacteria based on size, granularity, and fluorescence in a statistically measurable manner, using hydrodynamic focusing to align particles in a single file laminar flow for individual measurement. With fluorescence from autofluorescence of natural fluorophores in cells like aromatic amino acids, DNA, and RNA, or introduce fluorophores via fluorescent protein expression or conjugated antibodies, engaging with SSC or side scatter proportional to cell complexity, FSC, or forward scatter proportional to cell size, plotting SSCH versus FSKH to display complexity and size where each dot represents one cell. Using parameters like 78, a fluorescent probe marking dead cells, and EGFP, a fluorescent protein indicating transfected cell expression, yielding results with bottom left as unlabeled autofluorescent cells, top left as EGFP positive, transfected live cells, bottom right as 7 positive dead cells, and top right as double positive transfected but dead cells. Verifying transfection with immunocytochemistry examines the intracellular distribution of endogenous or transfected proteins in cell cultures, growing cells on sterile glass slides or glass-bottom dishes, since plastic dishes are unsuitable for fluorescence microscopy, fixing cells at desired density, permeabilizing membranes for intracellular markers, and incubating with primary antibodies targeting the protein of interest, detected via enzymatic color reactions or fluorescence with secondary antibodies, recognizing the primary antibody Fc region, fused to fluorochromes, using fluorochrome-labeled secondary antibodies from different species for multiple antigens, if primary antibodies are from distinct hosts. Validating specificity by omitting the primary antibody as a staining control to check for non-specific secondary antibody binding where residual fluorescence indicate non-specificity, and further tests like Western blot, affinity chromatography, or staining knockout cells or tissues. With multiple fluorescent labeling, including the nucleus with DAPI excitable by UV, the Golgi complex with G130 labeled by Alexa 633, the F-actin network with phalloidin labeled by Alexa 546, and EGFP present in the cytoplasm. Nucleic acid sequencing is the process of determining the nucleotide order in a DNA molecule, with modern techniques successfully sequencing the complete genomes of numerous organisms, including humans, evolving from early methods in the 1970s like chemical sequencing and Sanger sequencing, which advanced with fluorescent detection and automation to make sequencing faster and easier, leading to next-generation sequencing. Origins methods by the turn of the millennium that offer greater efficiency and lower costs. Chemical sequencing, now largely obsolete and replaced by Sanger sequencing, is based on chemical modification of DNA, followed by cleavage at specific bases. Starting with DNA strands labeled at the 5' end with radioactive phosphorus 32, separating complementary strands via gel electrophoresis, dividing the sample into 4 parts, each treated with a chemical to cleave at specific nucleotides or pairs. Formic acid depurinates adenine and guanine. Dimethyl sulfate methylates guanine. Hydrazine methylates cytosine and thymine, and hydrazine with sodium chloride inhibits thymine modification to target cytosine. Then, adding hot piperidine to each sample to cleave the sugar phosphate backbone at modified sites, adjusting reagent concentrations so each DNA molecule is modified only once on average, separating fragments by denaturing polyacrylamide gel electrophoresis to resolve even single base differences, and identifying them via autoradiography, deducing the nucleotide sequence from band positions across the 4 reaction lanes on the gel. Chain termination sequencing, known as Sanger sequencing, synthesizes a new strand from a template using DNA polymerase and a primer, adding chain-terminating dideoxynucleotide analogs, or DDNTAs, to 4 separate reactions, where DDNTAs lack a 3' hydroxyl group to prevent further phosphodiester bond formation after incorporation, halting DNA polymerase activity, with each reaction containing all 4 deoxynucleotides, or DNTPAs, plus 1 type of DDNTP, like DDTP-TP, or DDTP. So in the DOT reaction, synthesis stops randomly at thymine positions on the template, producing fragments of varying lengths corresponding to each thymine, with the DDNTP to DNTP ratio ensuring termination at all complementary positions, resulting in 4 reactions revealing the positions of each nucleotide type. DZTP shows cytosines, DDT shows guanines, allowing the full sequence to be reconstructed. Read bottom to top from the primer, which isn't visible, across the 4 lanes to form a sequencing ladder, resolving fragments up to 500 to 1,000 nucleotides reliably. Originally visualized manually via autoradiography with radioactive labels on primers, DNT is often DCPP, or DDNTS, using phosphorus 32, a beta emitter with a short half-life of 13.4 days, requiring strict safety measures and rapid sample degradation, or sulfur 35. An alpha emitter that's safer with a longer half-life of 87.5 days, but a weaker signal needing longer autoradiogram exposure. Automated fluorescent sequencing, accelerated Sanger sequencing to enable large-scale genome projects, sequencing 800 to 1,000 base pairs, running reactions in a PCR machine to amplify synthesis from less template LGBTQIA. Friendly PCR products can be used as templates too, advancing with the Kleno fragment of DNA polymerase first, replacing radioactive markers with fluorescent dyes attached to DDNTs or primers, with big dyes common DDNTP, analogs featuring 4 distinct fluorescence wavelengths to allow all reactions in a single tube, separating fragments by capillary gel electrophoresis, where shorter fragments reach the detector first, detecting via fluorescence spectroscopy with a UV laser exciting field. Gel and emitted light measured at 4 wavelengths by a detector, analyzing data computationally to produce a chromatogram or sequinogram, displaying the nucleotide sequence in 4 colors: red for thymine, green for adenine, yellow for guanine, blue for cytosine, validating accuracy by sequencing both strands to confirm complementarity. Chromatogram evaluation for sequence comparison notes that uneven incorporation of chain-terminating nucleotides can produce irregular peak shapes and height, while an SNP, or single nucleotide polymorphism, indicates a single base pair difference, such as cytosine in one allele and thymine in another. Genome sequencing through whole genome sequencing includes the map-based or clone contig method, which involves creating or using a pre-existing physical and genetic map of the genome before sequencing to guide the placement of sequenced DNA fragments within the genome, starting with physical mapping where large genomic DNA segments are cleaved. With a restriction enzyme like HindIA, separated by gel electrophoresis to produce a cleavage pattern known as a DNA fingerprint, and overlapping regions are identified by matching fingerprints to determine the fragment's genomic position, followed by genetic mapping that provides relative positions of DNA segments, noting that genes or regions in close proximity are more likely to be co-inherited than distant ones. Using multi-generational studies and DNA markers like RFLP or restriction fragment, link polymorphism for variations in fragment sizes, SSLEP or simple sequence length, polymorphism for variations in repeat sequence lengths, and SNP or single nucleotide polymorphism for single base pair differences to map gene distances. The shotgun method, or whole genome shotgun sequencing, offers a simpler, bottom-up approach requiring significant computational power. Performed without prior mapping for faster results, fragmenting DNA into small pieces of hundreds of base pairs, where smaller inserts allow plasmid vectors instead of specialized vectors to create a library of hundreds of thousands or millions of clones. Sequencing these clones and assembling overlapping sequence reads into contigs, then into the full genome sequence. Though misalignment can occur at repetitive sequences, as exemplified by Haemophilus influenzae in 1995, when Craig Venter and team sequenced its 1.83 megabase pair genome. Using this method, fragmenting DNA by sonication, separating it by electrophoresis, isolating 1.6 to 2 kilobase pair fragment, cloning them into a plasmid library, randomly selecting 19,687 clones to yield 28,643 sequences with reads of a few hundred base pairs. Reading about 1/3 of sequences from both ends of clones to aid assembly, totaling 24,304 reads producing 12,630,485 base pairs, approximately 6 times the genome size. 2. Ensure complete coverage, assembling overlapping reads into sequence contigs, and filling gaps by screening the library for clones bridging contig ends. Closing physical gaps with synthesized primers from unassembled contig ends hybridized to a new library, or using paired oligonucleotides in multiple PCRs with products. Sequenced coverage, or redundancy, reflects the number of sequenced base pairs relative to genome size to ensure full representation. Where ONX coverage doesn't guarantee all regions are sequenced due to random clone selection, leading to redundant sequencing of some areas and gaps in others. While 6 to 10x coverage is typical, covering 99.8 to 99.99% of the genome, with high redundancy increasing overlapping sequences to aid contig assembly. Paired end sequencing addresses the challenge of assembling sequence reads, especially with repetitive regions. By designing libraries with uniform DNA fragment lengths and sequencing both ends of each clone, providing known fragment length as distance information between paired reads. To improve assembly accuracy, genomic library construction includes cosmid libraries with insert sizes of 20 to 25 kilobase pairs. Combining plasmid and lambda phage vector advantages where appropriate sized inserts package into infectious phage particles, forming a library of recombinant cosmid vectors that infect bacteria, circularize, and replicate as plasmids. Screened by transferring bacterial colonies to nitrocellulose membranes, replicating them lysing with sodium hydroxide, fixing DNA chemically or by heat, and hybridizing radioactive or fluorescent DNA probes to identify target clones via colony hybridization. While artificial chromosome libraries handle insert sizes of 100 to 300 kilobase pairs, separating DNA fragments by pulsed field gel, electrophoresis, or FK, transforming via electroporation for higher efficiency than YACI libraries, and isolating their compact, supercoiled structure to simplify purification. Compared to YACI, or yeast artificial chromosome libraries, initially maintained as circular, plasmid-like DNA in E. coli, and linearized for larger inserts up to megabase scale, featuring bacterial and yeast replication origins, selectable markers, a multiple cloning site, and telomere and centromere sequences for stability and segregation in yeast. Screen most efficiently with DNA probe hybridization by transferring clones from agar plates to membranes in an ordered array, lysing, denaturing, and fixing them, with probes identifying clones carrying the target sequence on both membrane and plate. Next-generation sequencing, or NGS, encompasses high-throughput methods capable of sequencing many samples in parallel, known as deep sequencing or massively parallel sequencing, differing fundamentally from Sanger sequencing in chain synthesis and detection, requiring advanced robotics and significant computational power, introducing slightly more errors and producing shorter reads of a few hundred bases, but sequencing up to a million fragments simultaneously, dramatically increasing the efficiency of DNA-based studies, and reducing per-base costs to enable sequencing of the human genome in weeks or even hours, versatile enough for DNA, mRNA via RNA-seq, small RNAs through small RNA-seq, and ChIP-seq for sequencing DNA regions involved in gene expression regulation via chromatin immunoprecipitation, providing insights into cellular expression states and regulatory regions. Pyrosequencing offers read lengths of 100 to 500 nucleotides in solid phase with resin beads, or liquid phase with enzymatic preparation using aperase and X-nuclease, where solid phase emulsion PCR fragments DNA, amplifies it via PCR, and biotinulates it with primers, binding fragments to streptavidin-coated polystyrene beads with one DNA fragment per bead, emulsifying beads in an oil-water mixture where water droplets contain PCR reagent, sized to hold one bead each, transferring beads to a fiber optic plate with about 1.5 million nanowells of around 15 picoliters each, centrifuging them into wells with reaction components to amplify 100 to 500 base pair DNA per bead in isolated droplets, hybridizing single-stranded DNA templates with a primer and incubating with DNA polymerase, ATP sulfurylase, luciferase, aperase, adenosine 5'-phosphosulfate, or APES, and luciferin, adding DNTEs sequentially so incorporation releases pyrophosphate if complementary, which sulfurylase converts to ATP using APES, prompting luciferase to produce oxyluciferin from ATP and luciferin, emitting light via chemiluminescence, degrading unincorporated DNTS with aperase, tracking nucleotide incorporation in real time via light flashes, performing at about 10 seconds per reaction to sequence around 20 million nucleotides per plate, with software reconstructing the full DNA sequence. Though it's expensive and limited to 300 to 500 nucleotide reads due to small reaction volumes, shorter than Sanger's 800 to 1200 nucleotides, still sequencing bacterial genomes in 4 to 5 hours and eukaryotic genomes in a few weeks. Illumina or Solexa, sequencing fragments DNA, like an entire genome, into about 100 base pair pieces, repairs double-stranded DNA ends by removing sticky ends, adds a 3' adenine, ligates to a thymine overhang adapter, size selects and denatures fragments with sodium hydroxide, applies them to a sequencing plate with anchored complementary oligonucleotides as primers, uses bridge amplification to form clusters amplifying the target DNA, and sequences by synthesis with fluorescently labeled ddNTase, adding all 4 nucleotides simultaneously, each with a distinct fluorophore, cleaving and washing away fluorophores after detection to continue the cycle, sequencing the human genome in 7 to 10 days at reduced costs. Solid, or sequencing by oligonucleotide ligation and detection, uses oligonucleotide ligation for sequencing, offering lower costs but shorter reads less suited for genome projects, fragmenting the genome into 35 to 50 base pair pieces to form a fragment library, ligating adapters to both ends. Binding fragments to magnetic beads via primer hybridization with one DNA type per bead, amplifying DNA on bead surfaces via emulsion PCR. Denaturing templates, covalently attaching beads to a sequencing plate, and sequencing with 8 nucleotide oligonucleotides labeled with 4 fluorophores. Ligated in 7 cycles per template, repeated 5 times to map all bases. TSMs, or true single molecule sequencing, sequences single DNA molecules without PCR amplification, fragmenting DNA into 100 to 200 base pair pieces, denaturing them at 94 degrees Celsius, ligating single-stranded fragments to fluorescently labeled poly-A-tailed adapters, hybridizing fragments to oligo-T anchors on a chamber surface, mapping hybridization sites by fluorescence with a laser, cleaving and washing away fluorophores, synthesizing with DNA polymerase and fluorescent DNTs using oligo-T as primer, adding one DNTP type at a time with incorporation detected by fluorescence, followed by washing, photographing the plate after each step, forming up to 1 billion bases per day with a 0.5% error rate. SNR single molecule real-time sequencing sequences the human genome in about 15 minutes for around $100, using 1.5 to 2.9 kilobase pair fragments for greater accuracy, conducting reactions in tiny single-molecule chambers of 20 zeptoliters, each with one DNA polymerase fixed at the base. Using fluorescently labeled DNNPs, with the label on the phosphate group cleaved as pyrophosphate during incorporation. Detecting released fluorescent pyrophosphate in a nanophotonic chamber, with thousands of chambers per plate enabling parallel sequencing of long DNA fragments. Though homopolymer regions are challenging to resolve, relying on fluorescence intensity differences. Applications of sequencing span functional and environmental genomics, known as metagenomics, where sequencing enables the study of gene functions and microbial communities in their natural environments, while automated sequencing applies to sequencing PCR products and analyzing inserts from cloning experiments. And next-generation sequencing, or NGS, extends to whole genome sequencing, mRNA sequencing via RNA-seq, small interfering RNA, or siRNA sequencing, and chip sequencing, called ChIP-seq, which sequences DNA regions involved in gene expression regulation, isolated via chromatin immunoprecipitation using NGS methods, covering specific research areas, like comparing genomic versus mitochondrial DNA, differences across tissues, tracking developmental gene expression patterns to monitor gene activity during development, studying temporal DNA methylation, and histone binding patterns to explore epigenetic modifications over time, investigating non-coding RNA patterns for regulatory NNAs, analyzing single-cell DNA sequencing for individual cell genomes, advancing personalized medicine to tailor therapies based on individual genetic profiles, conducting transcript analysis to examine RNA populations, and profiling RNA types and quantities through RNA sequencing. Chip sequencing, or ChIP-seq, uses chromatin immunoprecipitation to isolate DNA regions linked to gene regulation, followed by NGAs to sequence these regions, providing insights into transcriptional control, while cDNA library construction generates a collection of complementary DNA, or cDNA, from nRNA, used for studying gene expression or cloning, often sequenced with NGAs or automated methods.